see my screen now? So cell cycle. What is cell cycle actually? So we know that cell division is important process in all the living organisms. Cell division is an important process. So during the division of a cell, what happened? DNA replication. What do you mean by DNA replication? Means DNA form its copy. That is DNA replication. So firstly DNA replication, secondly cell growth, that also take place during what? During the division of a cell. Whenever our cell divide, so at that time DNA replication, number one DNA replication, number two cell growth, that also take place. So all these processes, that is cell division or DNA replication or cell growth, hence have to take place in a, they should these three processes should occur in a coordinated way. There should be proper coordination in between these three processes to ensure correct division and formation of progeny next generation, that is progeny cells. So underline this one, this definition: the sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell, and eventually eventually divides into two daughter cells, that is known as what? That is known as the cell division. So what is the genome actually? You can write its term meaning. What is genome? So the entire set of genetic material. The genetic material is what? DNA or RNA. So the entire set of genetic material, that is DNA or RNA, present in an organism, that is known as what? That is known as the genome, right? So basically what I am saying here, the sequence of events by which cell duplicates its genome, means I am saying duplication of genetic material, that is DNA, and also synthesizes the other constituents of the cell, components of the cell, constituents of cell, and then eventually divide into how many daughter cells? Two daughter cells. So that is termed as cell cycle. So although cell growth is a continuous process, so DNA synthesis occur only during one stage in the cell cycle. Main important things that we have to focus, that is the phases of cell cycle. Phases of cell cycle. So, a typical eukaryotic cell is illustrated by human cells in culture. These cells divide once in, underline this thing. Typical eukaryotic cell divides once in every 24 hours. A typical eukaryotic cell divides once in every 24 hours. So, but however, this duration of cell cycle can vary from organism to organism and also from cell to cell type. This is important. Yeast, for example, yeast is unicellular or multicellular? It is unicellular. Okay. So it can, in case of yeast, can progress through the cell cycle in only about 90 minutes. So what is the duration in case of yeast here? For the complete cell cycle, 90 minutes. So that is given. So the cell cycle is divided into how many phases? Mainly two phases, interphase and M phase. M phase is the mitosis phase actually, right? So what is the M phase? Firstly we will understand here what is M phase. The M phase represents the phase when the actual cell division, M, you can remember in mind M for mitosis to understand. So M phase represents the phase when the actual cell division or mitosis occur. So it is the phase where the actual cell division or mitosis occur, that is M phase. And the interphase represents, very very important, interphase represents the phase between two successive M phases. So there is one M phase, there is other M phase. So it represents the phase between two successive M phases. Which one? Interphase. So what is interphase? What does it represents? Phase between two. Great. So next, it is significant to note that in the 24-hour average duration of cell cycle of a human cell, cell division proper lasts for about, it is very significant that 24 hour, which is the complete duration of the cell cycle of a human cell, but in that 24-hour duration, cell division proper lasts for only about half an hour. So, but interphase lasts more than 95%. This is very, very significant to note that interphase lasts more than, so that means interphase is taking nearly 20 nearly 95% of duration of complete cell cycle. Which one is taking? Interphase. Understood? So the M phase starts with the nuclear division. M phase start with the nuclear division, then corresponds to the separation of daughter chromosomes, that is karyokinesis. So what is karyokinesis? Karyo means what? Nucleus. Karyo means nucleus. So karyokinesis is what? Noted down here in this space. In the means terminology, meaning you have to write in NCRT, okay? So that easy. So division of the nucleus during cell division. So karyo is equal to, we have to split the word very easily. Karyo is equal to nucleus, and kinesis is equal to, kinesis is equal to movement or division. And this karyokinesis is what? This is the first step here for, first step for mitosis, followed by, after that, as it is mentioned here, M phase starts, mitotic phase start with the nuclear division, I told you, right? And it usually ends with the division of cytoplasm. What is what is cytokinesis? Division of cytoplasm. So we understood very clearly that our first one will be what? Our first one will be the karyokinesis. It is the, it is the M phase starts with what? Nuclear division, that is karyokinesis, and it ends with what? Cytokinesis. So the interphase, what is the other name of interphase? The other name of the interphase is the resting phase actually. It is a time during which the cell is preparing for the division, resting. If you are taking rest, so means you are not working, you are just preparing yourself for next day work. So same thing you can remember here. So resting phase, it is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication. In the resting phase, both, in the resting phase, both cell growth and DNA replication is taking place in the orderly manner, in a proper order. So it is the time during which the cell is preparing for the growth. And if anyone asks you that during this time what is happening, so number one, cell growth, number two, DNA replication. So interphase is divided into how many phases? It is divided into three phases. Three phases are, three phases are, number one, G1 phase. Number one is the G1 phase. Number two is the S phase. S phase is the synthesis phase. Okay. Next is the, am I clearly audible? Screen visible? If there will be any problem, you can let me know. Okay. I'm just explaining it. So G2 phase is the G2 phase, gap 2 phase, G2 phase. So G1 phase corresponds to the interval between mitosis and initiation of cell division. So it is what? It is the interval between what? Mitosis and initiation of DNA replication. It is the first gap phase of cell cycle. It occurs just after mitosis. Note it down so that it will be more clearly understood. In this space you can write more clearly. It is the first it is the first gap phase. It is the first gap phase of it is the first gap phase of the cell cycle. In the bracket you can write part of interphase. It is the part of interphase, and it occurs it occurs before DNA synthesis. It occurs before DNA synthesis. DNA synthesis occur DNA synthesis occur in which phase? S phase. We will discuss S phase. Okay. So this is occurring before DNA synthesis, means before S phase. So important thing to understand here, underline this: during G1 phase, cell is metabolically active and continuously grows, but it does not replicate its DNA. Metabolically active cell divisions are taking place, so it grows and but does not replicate its DNA. So cell growth is what? That is cell increase in size. So main feature of G1 phase is what? Cell increases in size, that is cell growth. But and continuously it grows, but it does not replicate its DNA. So S or synthesis phase marks the period during which the DNA synthesis or replication takes place. So DNA synthesis or replication take place in which phase? S phase. So during this time the amount of DNA per cell doubles. The amount of DNA per cell doubles. In which phase? So it simply means that if the initial amount of DNA is denoted as 2C, then it increases to 4C. Okay, means it doubles. So if I am representing initial amount as 2C, then it increases to 4C. However, there is no increase in chromosome number, that will remain as such. So if the cell had, so it means that if for example my cell have deployed, that is 2n number of chromosome at G1, then even after S phase also it will remain same. So the number of chromosome will remain the same as 2n only. So in animal cells, during the S phase, DNA replication begins in the nucleus and centriole duplicates in the cytoplasm. During the in animal cells, what happened? DNA replication began in the nucleus, obviously, because nucleus is there, and DNA is genetic material is inside the nucleus. Centriole. It is also a cell organelle in case of like a typical feature. You can write here below this. It is what? It is a cell organelle that I told you. So, a small cylindrical, a small cylindrical cell organelle found in most animal cells. In case of animal cells, there is a small cylindrical cell organelle which is called as centriole. What is its function actually? What is the function of centriole actually? Its function is cell division. What is its function? Cell division. Clear? No, outside. Why outside? Yeah, of course. Why? Because all the cell organelles they are present inside the cytoplasm only. For example, this is a cell, animal cell. Okay, here nucleus is there. So chromosome or your genetic material that will be inside the nucleus, whereas your cell is filled with the semi-fluid substance that is called as cytoplasm. That is called as cytoplasm. So all the cell organelles they are present inside what? Inside the cytoplasm only. Inside the cytoplasm only it will be present, whether it is centriole or mitochondria or anything that will be present inside the cytoplasm. So that's why they are mentioning like this. Centriole duplicates in the cytoplasm. Duplicate means to produce the copy, own copy. Now next comes G2 phase. During the G2 phase, the proteins are synthesized. So proteins are synthesized in preparation for mitosis while the cell growth continues. So proteins are synthesized in which phase? G2 phase. It is actually like G1 phase is actually growth. You can write for more better understanding in this space in the box. G1 phase is equal to, just to compile everything in a short note form, you can write in one line. G1 phase is equal to growth plus preparation plus preparation phase. Growth plus preparation phase in the bracket before DNA replication. Before DNA replication. Clear? Now there is very interesting point here. Very interesting point here to note, that is, there is one main feature of G1 phase. What is that feature? Actually, main feature of G1 phase is also that it consists of the checkpoint, G1 checkpoint. So this checkpoint ensures that cell is ready. You can include this, not given in NCRT clearly. So you can include this in this space so that you can clearly understand next paragraph also. That is checkpoint, or we also call it as G1 checkpoint or restriction point. It ensures the cell is ready to enter. Are you writing this point? also note down the points that mean easy points that I will discuss here. Okay, that also you note down, so that it will be easy to recall as a future reference also. You all terminologies will be clear to you. So, G1 checkpoint or restriction point ensures the cell is ready to enter to cell is ready to enter S phase. So, checkpoint G1 checkpoint or restriction point ensure the cell is ready to enter S phase. So, what is the outcome here? What is the outcome here? If all conditions like all nutrients or any type of signals, these are like the if everything is fine, these are correct, then at that case cell, what will happen, cell proceeds to S phase. If not if not if all conditions are not fine, if not, then it may enter, your cell may enter a resting stage called G naught phase. This is what you will study after that, so I am discussing, called G naught phase. Yeah, like all the nutrients, like that is the checkpoint actually. So actually, your G naught phase is what? That is the resting phase or quotient stage given in the NCRTC. This one, yeah. See, some cells in the adult animals do not appear to exhibit division. For example, heart cells and many other cells divide only occasionally as they needed to replace the cells that have been lost because of injury or cell death. Those cells that do not divide further exit G1 phase. This is important. Underline, those cells that do not divide further exit G1 phase to enter an inactive stage called as the quotient stage of the cell cycle. It is also called as what? Resting phase. It is also called as resting phase. So cells in this stage remain metabolically active but no longer proliferate unless called on to do so depending upon the requirement. I will discuss here in detail what it is. I told you that cell is metabolically active but it does not divide. It is also called resting phase, though the cell still perform normal functions. So what are the main features here? No DNA replication. Why? Because the cell does not enter S phase. Okay. So actually, there are some cells. More clarity I will give you in the space here. You can also write. This G naught phase can be temporary or permanent. It can be temporary or it can be permanent. So now here the question arises that what are the examples where this is like temporary? This is temporary. So first heading is our, it is temporary. This G naught phase is temporary in case of, it is temporary in case of liver cells or lymphocytes. Means they can re-enter cell cycle, but that is permanent. Next heading, that is permanent. This phase is permanent in case of neurons. They are nerve cells. And also cardiac muscle cells. Cardiac muscle cells. Cardiac word is related to our heart. So cardiac muscle cells, they remain in G naught for lifetime. For lifetime. time. So see, point which is written here, some cells do not divide. Some cells in the adult humans do not appear to exhibit division. Example, heart cells. Completed. So what is the importance of this G naught phase? It is explaining why some tissues like brain, your heart, they cannot regenerate easily. You might have heard that. So what is the importance of this thing actually? What is the importance of this G naught phase? What is its importance? So its importance is simply it prevents it firstly it prevents unnecessary cell division. Unnecessary cell division. Secondly, secondly, it explains. It explains. Yeah, I am writing here also. Importance it prevents unnecessary cell division. Are you writing in NCRT or on separate paper? If you are writing on separate paper, then that is also fine, or you can attach that paper in your book also later. It will also be helpful. And if you are writing in the notebook, then you have to keep that notebook properly by giving the name, key points and definition. So because proper understanding of NCRT, we have to understand this thoroughly. If we just keep on reading, then it is like cramming. So we have to be thorough in study to get interest. So explain why some it explain it. First point I told you, it prevents unnecessary cell division. Then second, what is the importance of G naught phase? Second, it explains why some tissues. It explains why some tissues like brain, I told you, nerve cell, that cannot regenerate. So like brain, your heart cannot regenerate easily. So in short, in the same box you can write in the same way that I told you to write in a box. In short, to compile that G naught phase, in short, make box like this and write there. G naught phase is equal to resting, is equal to a resting, comma, non-dividing, non-dividing stage. Okay, resting non-dividing stage. And here cell is metabolically active. Metabolically active. So it means what? It means that cell is active but outside the cell cycle. Cell is active but outside the cell cycle. But metabolically it is active. Completed? So your neuron and your cardiac muscle cells, they remain in G naught for lifetime. They cannot regenerate easily. This is the reason. So now we can proceed towards the now we can proceed towards what? M phase. So firstly we must know what is your mitotic phase. So you can write here one thing, mitotic phase or our M phase. That is what? That is the division phase of the cell cycle. It is the division phase of a cell cycle. right? And what are your what are your stages of M phase? So M phase consists of two stages actually. We can make in the form of a flow chart. It consists of two stages. Number one, karyokinesis. Karyokinesis is what? Nuclear division. It is nuclear division. Secondly, we have cytokinesis. Cytokinesis is what? Cytoplasmic division. So karyokinesis consists of how many stages? Four stages. Four stages are prophase, metaphase, anaphase, and telophase. So these are the four phases actually. So mitosis is also called as, we will study mitosis. Mitosis is also called as equational division. You know why it is called as equational division? Hmm. Hmm. Means like number of chromosome in the offsprings will be same as that of the parent. Irrational division. Great. So we are going to discuss here first phase, that is the prophase. Prophase of the karyokinesis. So here you have to make some short notes. So you take out a pen paper with you. Okay, okay, fine. So we are going to discuss main points, means what are the main changes that are taking place in case of prophase of karyokinesis in mitosis. What are the main events that are occurring in the prophase? Main events. First thing you have to keep in mind that it is the longest phase of mitosis. It is the longest phase of mitosis, prophase, right? So pro in the prophase, what happened? Number one heading, right? Chromosome condensation. Chromosome condensation. What do you mean by chromosome condensation? You know? Hmm. Packed together. So condensation of chromosome is what? Means the chromosome, they become condensed. Whenever you will see the term that the chromosome, they are becoming condensed. So it simply means what? It means the process where, like a chromosome, they are becoming like thick. I can say thick or short. Means they are just compactly packed. They are becoming thick and short. They are getting condensed. So condensation of chromosome, that is the first point here in the prophase. Second, nuclear changes. Second is the nuclear changes. You must understand that in the nuclear phases, nuclear membrane that disappear. Okay, nuclear membrane that disappear. So actually, these are the main things that we have to keep in mind. And now we can study once NCRT point. See what they are giving. Main things that you have to keep in mind from examination point of view that I will discuss here, and later when you will have more time after the exam, so we will again discuss the chapters. But here from examination point of view, I will tell you main changes. First point, tick mark this. Chromosomal material condenses to form mitotic chromosome. Chromosomal material, condensation of chromosome, first one. Then next is the next you just underline this one. Cells at the end of prophase when viewed under the microscope do not show Golgi complexes, endoplasmic reticulum, nucleolus, and nuclear envelope. These all will disappear. Nuclear changes. These all will disappear. What is Golgi complex? What is endoplasmic reticulum? They are nothing but these are cell organelles. So these cell organelles, they will disappear. But one point you have to keep in mind, that in the last phase is what? Last last phase here is the telophase. So in the telophase they will reform. And also underline this, nuclear envelope was disappeared in the prophase, but it, yeah, envelope that is covering it. So it will again develop in case of what? Last phase, that is the telophase. So what does it mean? It simply means that the events in the last phase are just opposite to that of prophase. They are just opposite to that of prophase. So nuclear envelope is also called as nuclear membrane. So that disappear. Envelope name itself indicating that is a covering. So in your basic events, you have to keep in mind that I told you: chromosome condensation, first one. Then next is the nuclear changes, nuclear envelope that disappear. These main points you have to keep in mind. So means at the end of, I'm discussing it. So at the end of prophase, chromosome are short, thick, and visible. Note it down. At the end of prophase, chromosomes are short, comma, thick, and visible. Okay, and also condensation point, nucleolus disappear. These all we discussed. So, firstly, you must understand what is centrosome before discussing the centrosome point. So, what is centrosome? Note it down. Centrosome is a small, non membranous cell organelle found in animal cell. It is a small, non membranous organelle present in animal cell. located near the nucleus, Loc- present in animal cell, small non-membranous organelle present in animal cell, located near the nucleus. It is located near the nucleus. It is made up of two centrioles. I am writing here, it is made up of two centrioles. Okay, fine. So what you wrote, it is made up of, centrosome is made up of, centriole is also what? That is also a cylindrical organelle found in animal cells. Okay. So it usually occur in pairs inside the centrosome. It usually centriole occur in pairs inside the centrosome. So what is the? I will repeat. Which one? Hmm. I told this centriole only. Centriole that occurs in pairs inside the centrosome. And also in the NCRT you can see this point, this one, centrosome, which had undergone duplication during the interphase. Duplication was in the interphase I discussed. So that began to move toward the opposite pole of the cell. That began to move toward the opposite pole of cell. So you know what do you mean by this opposite pole of cell? Yeah, it is simply the two ends of the cell. So centrosome, they begin to move toward the opposite pole of the cell. In which phase? Prophase. Each centrosome radiates out microtubules, which are called as the esters. So what are microtubules? You know what are microtubules? These are hollow cylindrical protein filaments. Hollow cylindrical protein filaments found in the cytoskeleton of eukaryotic cells. These are hollow cylindrical protein filaments found in the cytoskeleton of eukaryotic cell. Microtubules. Hollow cylindrical hollow cylindrical protein filaments found in the cytoskeleton of eukaryotic cells. Clear? Understood? So each centrosome radiates out microtubules, which are called as esters. So two esters together with the spindle fibers form the mitotic. There is a term, that is mitotic apparatus, that is formed here. Spindle fibers and esters. So whenever I am saying spindle fiber, so what is coming in your mind? Spindle fiber. Fiber means thread like protein structure. Yeah, these are thread like protein structures made up of mainly of microtubules. Thread like protein structures made up of microtubules. So a difference between spindle fibers and esters. So spindle fibers are what? These are thread-like structures made up of microtubules, whereas esters are what? They are star-shaped arrangement of microtubules radiating from centrosome. You can write here also. You will never forget. I'm sharing here a blank slide. In your page, you can write these two things. Difference. First thing you asked, that is spindle fibers. These are thread-like structures made of microtubules. Next heading you give formation. Next heading you give, that is formation. They develop from where spindle fibers develop. So they develop from the centrosome or centrioles during cell division. Can you clearly hear me? Can you see my screen? Okay. Spindle fibers are what? Thread-like structures made up of what? Microtubules. It developed from what? Centrosome or centriole during cell division. Next, what are esters? So they are, this is nothing but this is star shaped arrangement of microtubules radiating from the centrosome. Radiating from the centrosome. Complete this, then tell me. Star shaped arrangement of microtubules radiating from centrosome. Okay, fine. Okay, okay, great. So next is, as they are mentioning, that the two esters together with the spindle fibers form the mitotic apparatus. So what are the components of mitotic apparatus actually? Spindle fibers, and number two, esters. Number three, what can be here? Centrosome, or centrioles. Uh, They are also here. So in this way, prophase. Can you compile the prophase in 2 3 points? Tell me about prophase. Then we will follow. Mm-hmm. Okay. Great. So metaphase, the complete disintegration of when means your nuclear envelope that is completely detached. That is completely, yes, tell me. Yeah, yeah, I am just ending. I was also thinking to end it at 8:45. Yeah, 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 it was my previous plan actually to end it. So because I also have some other class. So the complete disintegration of the nuclear envelope marks the start of the second phase of mitosis. Hence, the chromosomes are spread through the cytoplasm of the cell. So by this stage, condensation of chromosome is completed. Yeah, condensation of chromosome that started in prophase. So, but the condensation of chromosome is completed in metaphase, and they can be observed clearly under the microscope. In which phase chromosome are clearly visible under the microscope? Metaphase. So this is the stage at which the morphology of the chromosome is most easily studied. Morphology means external appearance of the chromosome, that is most easily studied in this phase. Which phase? Metaphase.